In particular, the model is driven by increases in orientation variance and increases in contrast. And what we can see in that case is that we can explain across the electrodes, different electrodes in the 3 patients, that we can explain a large percentage of the variance in our narrowband recordings. So let me provide an intermediate summary right here. I've shown you how we need methods to characterize and parameterize power spectra, because then we can start separating gamma oscillations from broadband responses, and we can see that they have different stimulus specificity. And we can develop encoding models for this. And so what's important here as well is that the question is not whether there is any narrowband gamma for certain stimuli, such as does like one of these patterns induce any gamma or not, is not the important question to ask. The question is how we can explain observed variability between images. And image computable models make quantitative, testable, and interpretable predictions, and they can also be falsified and they can be built upon. We can say, like, this is what is explained and this is what's not explained. And we can take it also beyond the data that are tested. In this case, we tested it on a certain set of data, used cross-validation, but they can also be used on different images that we haven't shown yet, and see to what extent they explain that variability in new data sets. And using an image computable model, we found that orientation variance and contrast in the population receptive field predict gamma amplitude. And these features have been associated with gain control or suppression in neuronal circuits, and not necessarily with feed-forward processing. So these provide a testable, quantifiable prediction for the role of gamma oscillations in visual processing and what type of images will induce that. And I think that's very important for better understanding different diseases, because our typical approach comparing these signals leads to arguments as to whether one is better than the other. But that doesn't really help, because in photosensitive epilepsy, all these evoked potentials, neural sensory, and fMRI signals increase in amplitude. So if we think about what do different measurements encode, we can actually start learning a lot of new things. So let's apply this to photosensitive epilepsy. And let's see how gamma oscillations, for example, on epileptiform responses, whether they are driven by the same stimuli. Because I showed you in our data that gamma oscillations reduce. There's an increase in noise. And other people have found in the literature that epileptiform responses measured with EEG when patients with photosensitive epilepsy look at different images also reduce when there's added noise. And so the gamma doesn't only represent our data, it also represented a literature review for the last several decades where people have measured unit recordings, local field potentials, and MEG signals for different types of visual images. And so, for example, we summarize from these many studies how they drive gamma oscillations in epileptiform responses. And what we see is that gamma oscillations change in a consistent manner, for example, with increases in size. Gamma oscillations increase in power, increases in contrast, as I showed you, increases gamma oscillations. Multiple orientations decrease gamma oscillations. Changes from black-white to red-green luminance. Spatial frequencies. Increases to middle spatial frequencies, decreases to higher spatial frequencies, peaking around 2 to 4 cycles per degree. Grading edges don't really matter versus sinusoidal. We see the same pattern for epileptiform responses. However, if we summarize the literature on firing rates, it changes in a very different manner. For example, increasing in amplitude due to surround suppression for larger gratings compared to smaller gratings. And the fMRI signal also changes in a very different manner compared to